Thank you very much for the invitation to share with you some of our work regarding trained immunity, a process in immunology which is very important, we believe, for population health around the world and also in Africa. First of all, I would like to disclose that I am head of Division of Experimental Internal Medicine, Radboud University Medical Center, Nijmegen, the Netherlands, and at the same time, scientific founder of Train Therapeutics and Discovery. Why were we interested in this process and how did we discover it? Well, BCG, Bacillus-Calmette-Guerin, which is the only vaccine against tuberculosis, was developed approximately 100 years ago. It was developed at the Institut Pasteur by Albert Calmette and Camille Guerin, and it is still one of the most used vaccines around the world. However, something that is very interesting happened very quickly after the BCG was introduced in the population. For example, this is one of the first studies which was reported after the introduction of BCG in Sweden, and people have observed that mortality in children receiving BCG was decreasing spectacularly after the vaccination. In the population before BCG vaccination, this is the time before the discovery of antibiotics, approximately 11% of the children died of infections in the first 4 years of life. However, after BCG vaccination was introduced in Sweden, this percentage decreased to 4%, so more than 50%. That was not due to tuberculosis alone, because the tuberculosis death in this population was less than 0.5%, and it was due to other respiratory tract infections and neonatal sepsis that made Carl Näslund, who introduced BCG in Sweden, say that it appears that BCG provokes a non-specific immunity against other types of infection. This observation was made not only in Sweden, but also in other countries around the world, including in Africa. These are the results of 2 studies in Guinea-Bissau in West Africa, in which it has been observed that mortality due to infections decreased from, again, approximately 10% to 4%, whereas in Congo, in the same period, it decreased from 4 to 2%. So again, a beneficial effect of BCG of approximately 50% reduction in mortality. This data made the WHO ask a panel of experts to make an overview of the BCG protection against all-cause mortality. And this is the results of a study which was published in British Medical Journal in 2016. And it has observed that in many studies, in the vast majority of the study, BCG had a protective effect. Everything that you see on the left side of this line is a protection given by BCG. So it has been concluded that BCG most likely protects against all types of infections and not against tuberculosis alone. Additional studies have been done also in adults or in the elderly. This is a study published, for example, in South Africa in New England Journal of Medicine in 2018, when 73% less respiratory tract infections have been seen in adolescents revaccinated with BCG. And in the elderly in Indonesia and Japan, also between 70 to 80% less respiratory tract infections have been observed. How can we explain that? Can we first show this in a model of experimental human infection, that this is indeed the case, that BCG can protect against other types of infections? We have performed a study a couple of years ago in which we used as a model of experimental infection the vaccination with yellow fever vaccine. Yellow fever vaccine is a live virus, is an attenuated virus which infects the people but is not dangerous at all, and it can be eliminated from the body in approximately 1 week. And what we observed is that in the BCG vaccinated individuals, the viremia, the number of virus particles in the blood of the healthy individuals vaccinated with yellow fever vaccine, was significantly lower if individuals received BCG 1 month earlier. On day 3 and day 7 after the yellow fever vaccination, there were very few viral particles in the blood. But on day 5, when the viral particle numbers was the highest in the blood, the BCG vaccinated people had much less of the virus, meaning that BCG was indeed able to protect the individuals against this very mild viral infection. However, this is very important because this demonstrates that indeed BCG can be protective. Now, the question is, how does BCG work? How can we learn from that so we can use this information for further protection in the population? So usually the host defense mechanisms are divided in two parts, two main components, what it is called innate immunity and adaptive immunity. Innate immunity is what is happening in the first couple of hours and days after an infection. And there are especially what we call myeloid cells, macrophages, neutrophils, monocytes, which actually phagocytose, ingest bacteria, kill the infected cells, and then are able to usually eliminate the vast majority of infections. If that is, however, not happening properly, if the infection is too strong, then additional cells come into play, which are called lymphocytes, B and T lymphocytes, which are forming adaptive immunity that needs 10 to 14 days to develop, but is very specific against a certain microorganisms. And very importantly, they also build immunological memory, so they can remember the previous pathogen and then act accordingly the second time. We have always thought that innate immunity, which is non-specific, which does more or less the same thing every time that it's encountering a pathogen, lacks immunological memory and acts precisely in the same way every time it encounters an infection. However, what we have observed after BCG vaccination is that if we isolate these myeloid cells, innate immune cells from the body, after BCG vaccination, and we expose them either to Mycobacterium tuberculosis, for example, but also with non-related pathogens, such as Staphylococcus aureus or Candida albicans, they can respond stronger. This is the release of pro-inflammatory cytokines, important mediators of host defense, if the monocytes, myeloid cells from the BCG vaccinated are exposed to various bacteria and fungi. And we can observe that not only against Mycobacterium tuberculosis, but also against other bacteria and fungi, such as Candida albicans, these myeloid cells respond better after the vaccination with BCG, 2 weeks later or 3 months later. So how can we explain that? We can explain that by the fact that BCG acts at the level of the bone marrow, where the myeloid cell progenitors are present, and they are modifying that they produce cells which are better able to respond to infections. And we have done such study in which BCG vaccinated individuals were asked to donate not only blood, but also bone marrow aspirate. And we observed that also though their bone marrow cells work better. And this is the, let's say, the factory where all the immune cells are produced. And because of these changes at the level of the progenitors, these people have cells in the circulation which are coming from the bone marrow, which are better able to fight infections. So we were set also to try to understand the mechanisms through which these processes take place. And the mechanisms are taking place at the level of the chromatin, of the DNA of each of the innate immune cells. What is happening basically is that DNA in the normal state, if somebody is not infected, is very tightly bound in the nucleus. It's like a closed book which can not be read. It's very difficult to read a book which is closed. So especially in the case of naive macrophages, it's also very difficult for the gene transcription to take place. The gene transcription is very important for production of proteins which are important for host defense. If the macrophage is activated by an infection, then the chromatin, the DNA, is much loosely packaged in the nucleus. And then the transcription factors can bind to the promoters of the genes, and the genes can be transcribed. We can read basically the DNA to produce the mediators which we need for host defense. We always thought that these processes are going back to normal precisely in the same way after the infection is eliminated. However, what we have observed that there are certain chemical changes in the DNA, in the chromatin, especially methylation of the histones, which are proteins in the middle of the nucleosome of the chromatin, which remain methylated at the places which are necessary for host defense. Basically, the DNA is closed again like a book, but we put a bookmark at the right place. So the next time that we would need to read these instructions during the second infection, we can open the book of the DNA, we can open the chromatin precisely at the good places much quicker and much easier. In this way, what we have observed that during the second infection, the myeloid cells, after in the vaccinated individuals with BCG, can act quicker and stronger, and this enhanced immune response can defend us against secondary infections. This is how it is happening, that actually the myeloid cells of the innate immune system, also have a certain type of immune memory. Now, we were thinking also to use this process and try to protect healthy individuals against various types of infections, including COVID-19. The idea would be that, for example, if somebody has a low innate immune response, the virus can multiply very easily, that can induce a high number of viral particles in the blood, in the lungs, and so on. This could induce a very strong hyperinflammation around the body with a lot of symptoms, high severity of the infection, and unfortunately in some individuals with death. However, if we could boost the innate immune responses, we thought we could impede the virus to replicate. It would reduce the number of virus particles in the blood and in the body, which would lead to low inflammation, low symptoms, and survival. And because of that, we started first, this was even before the COVID-19 pandemic, with a study in the elderly, which was called Activate study, in which 200 elderly individuals who were at high risk of getting infections because they have been in the hospital with other types of diseases. When they went home, we randomized them to receive either placebo or BCG vaccination. And then we follow them for 1 year. And what we have observed is indeed that these individuals vaccinated with BCG had approximately 40% less infections than the individuals who received placebo. You can see it here in these curves. And the types of infections that had less, it was especially the respiratory tract infections, whereas urinary tract infections and abdominal infections, for example, were not modified by the BCG vaccination. It has to do most likely with the anatomical particularities of the lung, in which alveolar macrophages are innate immune cells which come into contact with the infection for the first time. So what we have observed from this study is that BCG vaccination induces production of genes which are important for host defense, which are improving the function of monocytes, which is inducing an increased defense against the virus and quicker elimination of all types of infections. At the same time, the plasma inflammatory mediators, which can be dangerous, are lower in these individuals. Due to that, we have started also a second study in which we wanted to see what is the effect of the BCG vaccination during the pandemic. And exactly like I showed you in the previous studies, we included in this study 300 elderly individuals who were randomized to receive either placebo or BCG vaccination. And during 1 year, we observed half a year. I apologize because here we had a little bit less time to observe the effects. We looked how much COVID-19 the individuals with BCG vaccination got compared with the placebo vaccinated individuals. As you can observe here, at day 90, 135, or 180, the individuals who received BCG, who are in red, they had significantly less COVID-19 compared with non-vaccinated or placebo-vaccinated individuals, meaning that indeed BCG was able to protect these individuals against infection. So based on this information, we can conclude that we can use probably live attenuated vaccines, which induce this improvement of innate immune responses that we call trained immunity in the first phase of a pandemic to decrease the impact of the new pathogen. These type of infections give a non-specific, this type of vaccinations give a non-specific protection. We cannot expect to have the same type of protection like, for example, with the specific vaccines that we use against the COVID-19, like the Pfizer vaccine, the Moderna vaccine, the Johnson & Johnson or AstraZeneca. They give a better protection because they are only against the pathogen. They activate the lymphocytes, which are very restricted in the way they respond. But even if we would observe a 40 to 50% protection by these attenuated live vaccines such as BCG, but also measles-containing vaccines, oral polio vaccine has been observed to have similar type of effects. We can mitigate the effects of a pandemic in the beginning and win time until we develop proper vaccines. So we don't need to have all the suffering and all the economic consequences of a severe pandemic as we had the last 1.5 years. Finally, I would like to show you that we can use this information, this knowledge, also to design new medicines, new drugs for other types of diseases. So, for example, another disease which is associated with a low response against of the immune system against a pathological process is cancer. In cancer, we also have the immune system which fails in its activation to eliminate the cancer. So we asked ourselves, would it be possible actually to design particles which activate these processes of trained immunity and use them in cancer? Because we know, for example, that BCG can be used in bladder cancer, but of course in bladder cancer, we can give intravesical the BCG, but we cannot do the same in other types of cancer because a BCG is a live microorganism and we cannot inject it in the people. So we wanted first to discover which is the component of the BCG which induces these effects. And this is the cell wall of Mycobacterium bovis, which is actually BCG. And you can observe all the components. So we studied which of these components induces these immunological processes. And what we observe is that muramyl dipeptide of the peptidoglycan component from BCG can induce the improvement of the function of the myeloid cells. You can see here cells which were exposed to MDP, this is the black bars, are better responsive to stimulation compared with the control cells. Then, with the help of colleagues from Mount Sinai, from the group of Willem Mulder and Zahi Fayyad, we have put together the technology, a nanobiological technology to incorporate this muramyl dipeptide in nanoparticles. And this is, we started a startup, Train Therapeutics discovery, in which myeloid cell-specific nanocarriers were built that integrate these components to activate trained immunity. These nanobiologics are HDL components, high-density lipoproteins, which we all have in the body, so they are completely non-toxic, and they are built from lipids and from apolipoprotein A1 protein, and then we build it that on the surface it has muramyl dipeptide, the component from the BCG, which is able to induce this trained immunity. Then we tested these nanoparticles. We have shown that they can induce this improvement of cell responsiveness. And then we assessed them whether they can be directed to activate especially the myeloid cells. And indeed, we radio-labeled them, and then we injected them either in mice, but also in bigger animals. And we observed that they go exactly to the organs which are very rich in myeloid cells, such as bone marrow and spleen. But at the same time, we have looked also at the tumor, and I will show you the data. what was happening with a melanoma, with a very severe and aggressive type of tumor, if we treat it with these nanoparticles. This cancer, this type of malignancy, melanoma, when injected in the mice, kills the mice very quickly because it is extremely aggressive. It kills the mice in approximately 2 to 3 weeks. So what we did, we injected in the mice the tumor cells, so they developed a cancer. We waited for 7 days, and then we started to treat them with MDP-containing nanobiologics. And what we observed, depending on the number of doses we gave, is that we diminished the growth of the bacteria with a higher number of the growth of the cancer with a higher number of doses of nanoparticles, of course. Thereafter, we have shown also that we can transfer this protection by transferring a new mice, the activated cells by bone marrow transplantation, and the transplanted mice were also more resistant to the tumor. In the end, we also combined these nanobiologics, which induce strain immunity, with checkpoint inhibitors, which are now used in the clinic for immunotherapy of patients with cancer. Well, checkpoint inhibitors are very inefficient in this type of very aggressive tumor. This is the growth of the tumor, and this is the growth of the tumor which is treated with checkpoint inhibitors. No difference at all. However, if we administer to the mice the nanoparticles which induce strain immunity, we see already an effect. But if we combine the nanoparticles with the checkpoint inhibitors, which are now given for immunotherapy, we observe a very significant reduction in the cancer. Actually, half of the animals had no cancer at all. There was a complete remission of the cancer, which is very spectacular in this type of model. So based on this, we try now actually to develop this new methodology, to develop this type of nanoparticles also for the use of patients, for the use of humans. So what can we conclude from all this, for all this information? Is that BCG vaccination, but also other types of live attenuated vaccines, such as measles-containing vaccines, MMR, or oral polio vaccine, can induce non-specific protection against heterologous infection. This effect is exerted on myeloid cells and their progenitors in the bone marrow, and they work through epigenetic and metabolic reprogramming of these cells. This can induce a major protection in case of infections and can be used, hopefully, in future pandemics for reduction of the impact of such new pathogen until new specific vaccines are developed. At the same time, we have shown that muramyl dipeptide from BCG can act and activate the immune system in the same way, and we can build nanoparticles that can be administered intravenous in animals and hopefully in the future in humans for treatment of other diseases which are characterized by a poor immune response, such as cancer. At the same time, what I do not have time today to go a lot into detail, there are also conditions in which these processes of trained immunity are activated in the wrong way, at the wrong place, and they can also induce autoimmune diseases, immune dysregulation, hyperinflammation, and so on. This has been shown, for example, in atherosclerosis. And atherosclerosis is a disease of the big blood vessels, and they are characterized, of course, as we know, with very severe complications such as myocardial infarction, brain attacks, and so on. So we have seen actually that there are components in our blood, especially some types of lipoprotein, oxidized lipoprotein, which can induce this activation of immune cells in the same way as pathogens, as bacteria do. Of course, this activation of the immune cells in the absence of an infection is not appropriate, and it's activated too much the immune system, and that can lead actually to the production of a lot of inflammatory mediators such as pro-inflammatory cytokines, and this can lead actually to a persistent inflammation in the blood and in the heart of these patients. This is what is happening actually with the cells which are exposed to these oxidized lipids from patients with atherosclerosis. They respond much stronger to any type of stimulation, as you can see here in the black bars, to any type of stimulation they are exposed to. Well, unfortunately, that can lead thereafter to a chronic inflammation in the blood vessel, and that can lead actually to production of activated cells in the cell wall, in the wall of the blood vessels, with production of pro-inflammatory cytokines, with formation of cells which are important in wall inflammation, and that can lead to myocardial infarction and other complications. So we thought ourselves, would it be possible that something like that can happen also in humans? And indeed, there are situations and there are important transition taking place nowadays in the whole world, but also on the continent of Africa, which increases the danger of these processes. We have done thus a study in Tanzania with colleagues from Moshi, from the Kilimanjaro University Medical Center, in which we have studied the immune response of people living in the rural areas around the Mount Kilimanjaro, and compare their immune responses and systemic inflammation to that of the people living in a city, in the city of Moshi, which is a city of approximately 200,000 inhabitants. And what we did, we isolated blood from these healthy individuals. We exposed them to different types of stimulations. And what we have observed is that the cells of the people from the town, from the city of Moshi, responded much stronger with production of inflammatory mediators compared with the people in the rural areas. You can see here everything here, which is a little bit violet, is increased, and it's higher in the urban population. Basically, the explanation for this observation was the presence of various metabolites in the food and in the blood thereafter of individuals, which differed between people living in the villages and eating a traditional diet compared with people living in the town, and who ate a much more westernized type of diet, including McDonald's type of food and so on. So what we have observed, basically, that the metabolites from the people eating a traditional diet, they had a much more modulatory function on the immune cells. They took care that the immune cells respond properly to all type of pathogens. Whereas the diet, the metabolites from the diet, people living in the cities like we do in the Western world, but what is happening now also in cities in Africa, those metabolites stimulated very strong the inflammation, unfortunately, in the blood of the individuals eating that type of diet. And that is likely, actually, that during the rural to urban demographic transition associated with changes in diet around the world, there is an increase of the pro-inflammatory profile, and thus most likely this is responsible for the increase in the metabolic diseases, in the increase in atherosclerosis, in cardiovascular diseases, all around the world, including countries in Africa, but also Southeast Asia and so on. This will lead actually to new challenges for healthcare, and we would need to tackle this problem in the future. In case of the train therapeutics and discovery that we are developing are also nanoparticles that inhibit these processes of inflammation and train immunity, which can be inappropriately induced by incorporating in the nanoparticles also drugs that inhibit and mitigate the inflammation, and then they are taken up by the myeloid cells, which are producing these inflammatory mediators, and thus we can downregulate it. So we can observe here in models of organ and systemic inflammation in the mice that if we administer them these nanoparticles, that we engineer the release of inflammatory mediators, the inappropriate release of inflammatory mediators in these models, is downregulated and it goes back to normal, like in the healthy mice. You can see here, these are the healthy mice. This is the increase in the inflammation when we induce this in these models, and we can return it back to normal after the administration of these nanoparticles. So finally, the point that I wanted to make is that the myeloid cells have very important components and capacity to modulate their function through chemical changes at the level of their nucleus. At the level of their chromatin, there are either methylation, acetylation, and other types of chemical changes that can either promote the function of the myeloid cells or inhibit their function. By modulating these processes, we can also treat different diseases. For example, in diseases which are characterized by too low immune responses, such as severe infections or cancer, we can build up drugs such as nanoparticles incorporating certain components for BCG, which can improve the response of the immune cells, and we can improve the elimination of the pathogen and treatment of the cancer. In other diseases, such as inflammatory and autoimmune diseases, in which the immune system is too strongly activated, we can also build this type of nanobiological therapy to downregulate the immune system and to treat these very severe inflammatory processes. Finally, of course, I would like to thank the people from Radboud University Medical Center, who are from our group, and from Mount Sinai in New York and Technical University Eindhoven, and also all the colleagues who contributed, and they are working together for the success of Train Therapeutics and Discovery, and also other collaborators around the world, which helped us in our academic projects. Thank you very much.